All right, we are on chapter 7, section 4, and we're talking about dietary lipids. So when we talk about dietary lipids, we kind of talked about this before, right? We talked about fats being solids at room temperature and oils being liquid. And so when you think about fats and oils, if you just kind of think about what state of matter they're in when they're sitting on your kitchen counter, you can figure out what it is. So the fats inside of meat, like steak, that's a fat. Lard is an example of a fat, and butter, right? If you just put butter out on the counter, it's soft, but it's still a solid. Oils, on the other hand, are liquids, right? There's soybean oil, there's vegetable oil, there's peanut oil, right? All of those are derived from plants. So whether they are fats or oils, they all belong to a general class of lipids. And that general class of lipids are hydrolyzable, and they're called triglycerides. So tri meaning 3, and glycer refers to glycerol. And we're going to draw that, and I'll show you what that is. But a lot of times what you're going to be asked to do is figure out, look at the structure. Is it a fat or is this an oil? And so all you have to do is look at the physical state of the triglyceride, because they're both triglycerides, and see, are they a solid at room temperature or are they a liquid at room temperature? And that's going to tell you what it is. And so when we look at a dietary lipid, we said it's a triglyceride. So what's the 3? Well, the 3 is I have 1, 2, 3 fatty acid tails. So that's my tri. Then glyceride, the triglyceride comes from glycerol. Glycerol is a carbon, right? And it's bound to hydrogens. It's got a hydrogen up here. It's got a hydrogen down here. And then it has hydroxyl groups. So if you link this to a fatty acid tail, remember what a fatty acid tail is. It's a hydroxyl, double bound here, and then you have your really long chain, right? So what happens is that you're going to do that, and you'll do the same thing, so on and so on and so on, right? And I'm not drawing it because I'm kind of out of room. But what's going to happen is you're going to form water, right? And I guess I should have drawn that. I should have drawn the hydroxyl the other way, right? Because it's not actually the carbon that's attached to the hydrogen. It's the carbon attached to the oxygen, right? And an OH and a carbon, OH and a carbon, right? So what happens to link these things together? Because you have to link these together in order to make a fatty acid. And so what happens is you actually release water, right? So you take a hydrogen from one and the hydroxyl from the other, and then you connect them. So this carbon will jump over and make a bond with this oxygen, and you pick up where you left off. So you had a carbon, oxygen, carbon, carbon, oxygen, and then carbon. And that carbon is right there in the carbonyl carbon. Okay, so that's how you make a triglyceride. If I could erase some of this. Okay. Ooh, I made a mess there. All right, there we go. So if we look at our triglycerides, they're structured very differently. So if we have a fat molecule, what you're going to see is that you only have 1, maybe 2 carbon-carbon double bonds. It's that carbon-carbon double bond that's going to change the molecule. So mainly inside of a fat, mainly we're saturated. We're saturated with hydrogen. So we don't have so many carbon-carbon double bonds, right? But look at an oil. Here, if I look at an oil, oh my goodness, look at all these. If I could circle them. Look at all these carbon-carbon double bonds. Because of all the extra carbon-carbon double bonds, this is an unsaturated fat, right? And so all of those carbon-carbon double bonds are cis, right? They're all going in the same direction. And they're going to make basically kinks inside of the hydrocarbon tail. So if you compare the two, right? Here, this is our like main carbon backbone. What you'll see is they line up pretty well together. But if you look over here in our oil, you see you have a lot of changes. You have a lot of kind of weird configurations. And so they don't line up one on top of the other very well. And so that's going to change their interactions, and that's going to change their states of matter. So if you look at the fat, right, and the fat model, in the fat, this is our saturated. So minimum, so 1 or 2 number of carbon-carbon double bonds, right? So you're mostly saturated. What you're going to see is that those carbons are very close to one another. And so the closer they are to one another, the more you can disturb the electrons within the 2 chains. And remember, what did we say that disturbing electrons, that force is? We said that is London forces, right? When you can just disturb the electron cloud and create that temporary dipole moment, that's a London force. And so the more they interact, the more of a London force they're going to have to hold them together. And so the tails of the fatty acids inside of a fat interact with one another. They have strong London forces, because London forces in general are weak, but they have a lot of them. So the mostly straight chain tails allow for more contacts. The more contacts you have, the more attraction you have. And so therefore you have a solid or a semi-solid, depends. But they're more solid. If you look at, on the other hand, the oil molecule, right? Oil is unsaturated. It has many carbon-carbon double bonds, right? And so it is polyunsaturated. Poly meaning more than one chain. So each of the chains, 1, 2, 3, is unsaturated. So all chains, all fatty acid chains are unsaturated. So these carbon-carbon double bonds, if you look at that backbone, if you kind of follow it along, it makes kind of a wave inside of the molecule. And so you can't line them up one on top of the other. They're too bunched up. And so they have less surface area contacts. The less surface area contacts they have, the fewer London dispersion forces they're going to have. They can't stack together very nicely. And so if you have less London forces, that means that the hydrocarbon chains can move around and flow more easily. And so that greater motion, that greater molecular motion, doesn't stack the tails. And so they end up being a liquid at room temperature. Okay, so let's try this one. Draw the skeletal structure of a saturated triglyceride with 18 carbons on each tail. Well, how are we going to do that? A triglyceride. Let's see. Let's look at this and explain things first. A saturated, what does saturated mean? Saturated means no carbon-carbon double bonds, all hydrogens, okay? Now, a triglyceride, tri meaning 3, so you have 3 fatty acid tails, and the glyceride is the glycerol backbone, right? So we said that was our 3 carbons, and this was 2 hydrogens, that's 1 hydrogen, that's 2 hydrogens, and then they each have the OH, right? Okay, so that's kind of what we need to get out of the information and then draw it. So I like to start drawing with my glycerol backbone. So carbon, 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 and you can draw your hydrogens. Okay, now I know that I'm going to make a bond with oxygen. In order to link this here with a fatty acid tail, one fatty acid tail, right, is going to be like this. And then on this one, we're going to have a double bond oxygen. So how do we link it together? We got to get rid of this and we got to get rid of that, because that's going to turn into water. So I've got my CH2C, and then I'm going to come over, pick up my oxygen here, and then go into my carbonyl. So I'm going to go carbon oxygen, and then it's going to go into my carbonyl carbon. Let me move this. Okay, I'm going to go into my carbonyl carbon. which I'm really gonna have to move that too. All right, let's see. Move that up here. Okay. So I'm going to go into my carbonyl carbon, which means I need a carbon-carbon double bond. Now, this needs to be how many carbons long? 18 carbons. So your carbonyl carbon does count as a carbon. So when you're counting by drawing, you need to do that. But what you have to remember is that when you're really counting carbons, the carbon that's farthest from the carbonyl carbon is really carbon number 1. So we're going to draw it from the glycerol end, but we'll have to go back and number it differently. So this is like carbon, this is a carbon. So that's like our first carbon, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, I'm running out of room, 17, 18. I got too close in there. I'm sorry. So this is really carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. And that's how many we should have, 18 carbons. I really scrunched it up over there. So what you just have to do is exactly the same thing for all of your other fatty acid chains, because we said it's a triglyceride. So we're going to go up, going to make a carbon-carbon double bond. So that's my first carbon. It's not really my first carbon, but it's my first carbon in the drawing. 1, 2, 3, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. I kind of spaced that one out, I think, a little bit better. Go back and double check and make sure that you put the correct number. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Now be careful, because on an exam, I will give you 1 more carbon and 1 less carbon as an option. So you need to make sure that you know that this carbonyl carbon is actually a carbon that you count. Otherwise, your count will be off. Okay, so let's do the same thing. We put a carbonyl carbon. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. All right. Phew. All right, so let's look at one more kind of fatty acid containing molecule, and that is called a phospholipid. So it's going to have a phosphate group and it's going to have a lipid. So phospholipids do have that same glycerol backbone, that 3-carbon backbone, and it does have fatty acids linked to it through that same ester bond, right? That's this ester bond. That's the ester bond right there. So it has the glycerol, it has the ester bond, but what's different is that a phospholipid only has 2 fatty acids. On that third one, instead of having a third fatty acid tail, what we're gonna have is a phosphate containing group. And so a lot of times that phosphate containing group is polar and big. And so now we have this opposite things going on, right? We have a very non-polar fatty acid tail, and we just stuck on a really, really big polar phosphate group. So now we're very amphipathic. Oof. All right, so what does this look like? Well, here in green, right, this is our fatty acid tail, just like what we've been drawing in the past. The pink is our glycerol backbone, right? We have carbon, 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 carbon. Now, you have attached to 2 of those ester bonds that are linking your glycerol to your fatty acid. But on the last one, we still have an ester bond, except this ester bond is between the carbon in the glycerol and, a phosphate. This is where the phosphate group comes from. So our phosphate group contains negatively charged oxygen, and we have a positively charged amine group. So this head group is charged. right? So it is polar. This tail region is non-polar and uncharged, right? So this is amphipathic. This is another example of that. And so a lot of times you'll see them in cartoons as just the phosphate head group, right? Those two are the same thing. And then the fatty acid tails, and they just kind of do them in a cartoon version. They're just a wavy tail, right? So there are a lot of different kinds of phospholipids depending on kind of the junk that's attached here. And they all share that similar structure where you have that glycerol backbone, those 3 carbons coming down, and the 2 fatty acid tails going out, and then the polar phosphate head group. So that's all common. But the phospholipids behave differently than our triglycerides. Our triglycerides have different shapes inside of the cell. When we look at our phospholipids, the phospholipids have a big head group and then kind of narrow down with that tail. And so we call them a almost cylindrical shape, right? With a big head group. And so you can't really form a micelle with that. It just doesn't work very well. The head group's too big for the tail. And so what happens is that they form what are called bilayers. So if you've taken biology, you know that the membrane of a cell is a lipid bilayer. Well, what does that lipid bilayer look like? Here it is, right? All of our polar head groups line up together. All of the fatty acid tails line up together. But think about a cell sitting in a basically aqueous environment. Aqueous means there's water. There's water all over the outside of a cell. Well, you know, the same's true about the inside of the cell. The inside of the cell is all water as well. And so those head groups are hydrophilic. They like water. And so they're going to line up. But what's going to end up happening is all these tails are going to be exposed to the inside of the cell, and they don't want to interact with water. So if this is your head group, and here's another one, right? This is your polar head group, and these are your fatty acid tails. They're going to end up lining up this way so that these fatty acid tails can interact with one another instead of interacting with the water. And so that's why we call it a lipid bilayer. Here's one layer, here's the other layer, okay? And so the polar head groups ended up being directed out into the surrounding aqueous environment and the aqueous interior of the cell. So the head groups, water's out here and water's down here inside of the cell. And so it protects the ends from having to interact with water. And so this is really the foundation of all of the cell membranes inside of every single one of our cells, right? So that's how you make a cell membrane, but you have other things inside of your cell membrane. Inside of your cell membrane, you have proteins that are gonna do jobs for you, like pump things in and out of the cell. They all have functional characteristics, like maybe they're receptors. They have all kinds of different jobs, but they're to connect information from outside the cell to inside the cell, right? And so you're going to have proteins. You're also going to have carbohydrates. Carbohydrates sit on the outside of the cell and are signals. Remember, we talked about these when we were talking about the blood groups, when we said that if you're A, you have 4 different carbohydrate pieces inside of your chain. If you're O, you only have 3, right? They're signals. And so if you think about how this works, it's not a rigid structure, the cell membrane. It's fluid. And I know that sounds ridiculously crazy because it's a solid, but it really is a fluid. And so the reason for that is you can kind of think about them like the proteins and the carbohydrates that are sitting on the outside. They're kind of like icebergs. And so they can drift around inside of the phospholipid bilayer and they can move. And so they call it the sea of lipids. And then you have these icebergs of proteins and carbohydrates that can move around. So the lipids are actually free to rearrange themselves. They have to stay in this conformation, but they can move like left to right and front and back. They can't move up and down, but they can move in 2 dimensions. And so here's kind of a diagram of the cell, right? And so you have all this green, right? You have a phosphate head, and there's the fatty acid tail. And here's on the other layer, phosphate head, fatty acid tail, right? And that's what makes up your phospholipid bilayer. You have your carbohydrates that stick out, but they can actually move around this way and that way, right? You also have proteins here that are like channel proteins that can pump things into or out of the cell, either way. And you have a few other things inside of the cell membrane. You actually also have cholesterol inside of the membrane. And the cholesterol is really, really important. It's in a class of molecules called steroids. And steroids are technically lipids, but they contain rings. And those rings are fused. And so all steroids will have this base structure that's called a steroid nucleus. And so a lot of different things can be attached to this nucleus, but they're all going to have this basic form. And we know, right, steroids look very, very different than the fatty acids and the triglycerides that we've talked about so far, but we classify them all together because they're non-polar in nature. So they behave very, very similarly. And they have very, very important functions in the cell. So we know about all the different steroids inside of our body that can control sexual development, like testosterone and estrogen. We know that there are some that help us to digest our foods, so help to break down fats inside of our foods. That's bile acids. And so they're very, very important. Cholesterol is one type of steroid, and cholesterol is important inside of our cell membranes. You do need some of it, not too much, but you do need some of it. And so cholesterol will actually wedge itself inside that phospholipid bilayer so that the hydroxyl group right here is going to protrude out into the extracellular environment. So this is going to be sticking outside the cell, right? And the rest of the molecule is nested inside the membrane. And this is inside the membrane. And you notice that the rest of the molecule is nonpolar. Oh no, what happened? Hang on. All right, trying that again. So the rest of the molecule sticks inside of the membrane, and that's the nonpolar part. And so that makes sense because it's trying to get away from the water. So if you have a low amount of cholesterol, the cholesterol will like slip inside in between the hydrocarbon tails inside of your membrane and can increase fluidity of the molecule. But if you get too much, it'll actually have the opposite effect, and it'll make the cell membranes very, very rigid. So it's an important balancing act for how much you're supposed to have inside the cell. All right, I think that's it. Yes, let's go do some practice problems. Oh. All right, so here are our practice problems. So we're going to start with our 7.4 dietary lipids. So in terms of their attractive forces, explain why fats are solids, but oils are liquids under normal conditions, despite the fact that they are both triglycerides. They are both triglycerides. So let's talk about their structure first. So fats. What do we know about fats? They are mostly saturated, saturated, so they have a minimal number of carbon-carbon double bonds, right? So if they have less carbon-carbon double bonds, we're going to say in the fatty acid tail, right? Because that's where they are. Then they're going to make more surface contacts. They make more surface contacts with the other tails. So why is that important? Well, the more contacts you make, the more London forces you get. What do more London forces tell you? More London forces tell you that they're going to be attracted to one another. The more attraction you have, the stronger the interaction. So you're going to have to in order to separate them, right, you're going to have to raise that temperature in order to get them to separate, to become a liquid. So they're going to stay a solid at room temperature. So this is solid at room temperature. RT is room temperature. Okay, oils. How are oils different? YLS, oils. So these are unsaturated, right? Unsaturated, we have lots of carbon-carbon double bonds inside of our fatty acid tail. So why does that matter? Well, because you have all these carbon-carbon double bonds and they're in the cis orientation, right? Every time they do that, they're gonna put it like a kink into the molecule. And so you'll have less surface area contacts. If you have less surface area contacts, you're going to have fewer London forces. So it's going to be easy to separate them. So at a low temperature, they're going to become a liquid. So they're a liquid at room temperature. They are liquids. Okay. So always think about that interaction between the two. Excuse me. Considering their chemical structures, why are melting points of oils lower than that of fat? This is what we just talked about, right? So which one's going to be lower? Oil has a lower, I'm just going to put MP for melting point. Why? We just said it takes less energy. So it takes less energy to and when we say energy, heat, right? Heat is temperature. To move our loosely packed tails. Right? Remember, these are the ones that are unsaturated. So it doesn't take as much energy to move them because they're not so packed together. Fats have tails that are more packed together. So melting point's higher, right? And so you kind of have to remember is how those molecules are interacting. All right, so this next one wants you to go back and pull a figure, and we're going to talk about that figure in a second. One of the triglycerides found in coconut oil contains 3 fatty acids with the carbon designations of 10 to 0, 8 to 0, and 12 to 0. Refer to, my screen sharing is paused. Oh. Why did I pause the share? Oh no, I wonder if y'all saw it or not. Go back and do it again. Okay, let me come back here. All right, so I think I wasn't sharing, but okay. So the very first one, just so you can see what I'm writing. So in terms of our attractive forces, why are fats solids, but oils are liquids? Fats have that smaller number of cis carbon-carbon double bonds in the tail, right? And they make more surface area contacts with the other tails. So as I was talking, I'm so sorry. I don't think I was sharing. I make mistakes too. All right, so our oils are unsaturated, right? Lots of carbon-carbon double bonds in that fatty acid tail, and they're in that cis orientation, and so that's what makes kinks inside of it. So we have less surface area contacts, so fewer London dispersion forces, making them liquids at room temperature. And it's a good thing I wasn't drawing anything. So if you just kind of want to jot this down, right? Consider the chemical structure. Why are melting points of oils lower than those of fats? Oil has a lower melting point because it takes less energy, less heat to move our loosely packed tails, our unsaturated fats, right? These are more packed together, and so they have a higher melting point, right? So fats, right? Fats have those tails that are more packed, so they're solid. Okay, now if we can go back here. Now, I'm sharing and I'm recording. Here we go. One of the triglycerides found in coconut oil contains 3 fatty acids with the following carbon designations: 10-to-1, 8-to-1, 12-to-1. Refer to section 4.5 for a review of fatty acid carbon designations. All right. The fatty acid with the carbon designation 8.0 appears on the middle of the 3-carbon glycerol. Draw the triglyceride. Okay. What does this designation mean? So I went and pulled the table from section 4.5. You're welcome. I know. And so whenever you're looking at these structures, here's the designation, right? This is 16-to-1. What does that tell you? That tells you that there are 16 carbons, and in those 16 carbons, you have 1 carbon-carbon double bond. Well, where is that carbon-carbon double bond? So you're going to call that omega-7. Omega-7 is going to be at the 7th carbon. You have the carbon-carbon double bond. So remember, when you count, be very, very careful. You start numbering at the carbon farthest from the carbonyl. Okay, so here's my carbonyl. That's my carbonyl carbon right there. So it's 1, 2, 3, 4, 5, 6, 7. So do you see here? That's why we call it an omega-7. Then 8, 9, 10, 11, 12, 13, 14, 15. Now that carbonyl carbon counts, and that is 16. Okay, so that's a 16 carbon fatty acid, and it has a one carbon-carbon double bond at carbon 7. Look at your next one, next example, oleic acid. This one has 18 carbons. It has 1 carbon-carbon double bond, but this time it is at the 9th carbon. So here's my carbonyl carbon. I'm going to start farthest away. 1, 2, 3, 4, 5, 7, 8, 9. So 9 is our omega-9. 10, 11, 12, 13, 14, 15, 16, 17. And our carbonyl carbon is 18. Okay, now we need to go and draw one. Now that we kind of remember how they're structured, we need to go and draw one. So we have 3 fatty acids. They are 10-to-0, 8-to-0, 12-to-0. So because they're all 2-0, this is telling me I have no carbon-carbon double bonds. The 8-to-0 is in the middle. So it doesn't matter what's on top or what's on bottom, because you can flip the molecule and it won't make a difference. But that 8 chain has to be in the middle. So if this is a glycerol, right? It's a triglyceride. So start with your glycerol. So our glycerol is going to be C, C, C. That's our backbone. So we have 2 carbons, 2 hydrogens attached to that carbon, 2 hydrogens on that one. Then we're going to have them bonded in an ester bond to our fatty acid. So that's going to be our carbonyl carbon. We're going to do a carbonyl carbon, carbonyl carbon. So this right here is carbon, counts as a carbon in the chain. So I need to make this carbon, let's do this one as 10, right? So this would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. But really when we count it, we would go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I always like to double check and make sure that I've made it the correct length. Okay, so my next one has got to be 8 long. So I'm starting here, that's 1 as my carbonyl carbon, 2, 3, 4, 5, 6, 7, 8, 9, 8. Let's count 1, 2, 3, 4, 5, 6, 7, 8. And that is correct. Okay. Now I got to do my last one, which is 12. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. and see if I did it right. And if you did it perfectly, your 12 should be longer than your 10, you know, that kind of stuff. But I'm not always a perfect artist. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And we do. Okay. So that's how we draw a triglyceride. Okay. What is next? So now we're going to talk about our attractive forces within the cell membrane, right? So how will phospholipids arrange themselves when they are put in water? Remember, we said that they can't form that micelle, and so what they're going to end up forming are lipid bilayers. So they will form 2 layers, right, called, I'm going to leave myself some to draw, called a lipid bilayer, right? Bi meaning 2, with the, how's it going to be arranged? With the non-polar tails, right, facing each other, and the polar heads facing, we'll say facing water or facing out, right? So if this is our polar head, that's our non-polar tail. Head and tail, head and tail, head and tail. So then we want a bilayer. So we're going to have to do the same thing, and the tails have to be facing one another. So these are our non-polar tails. This is our polar heads. Okay, so that's how they kind of look. All right, so let's see. Oh, and that was it. There's only 2 questions from that section. I can't believe there's only 2. Was it really only 2 questions? It was indeed. All right, y'all have a great rest of your day. And sorry about my error.